Okay, so I think we'll move on to our next speaker, if I may, who's Helen May Samira, who's come from the University of Mainz.
Good morning. Can you all hear me? I'm a lot shorter than Phil, so I'm going to have to bring this down. So many of you know me, but I've realised there's lots of new faces. So my name's Helen, and I started with Phil many years ago, and I now run a research lab in Germany, and I'm trying to understand what causes BBS on the cellular level. So we're very much a basic research lab, but trying to understand what's happening in the cells of patients that have BBS. I'm here with two of my lab members, two students that are sitting in the back. They're just waving their hands. And the project that I'm going to tell you about today is very much a project that they're involved in, and they are going to help me explain it to you. And also, like I said, hopefully you will all be very excited to help us, because I'm basically here today to say that we really, really need your help. Before we get started, though, just as a reminder to everyone, I'm just going to remind you what cilia are, because I know that Phil has mentioned cilia a couple of times, and explain to you why we need your help. So, as you all know, our body is made up of lots and lots of different cells, and many of these cells come in different shapes and sizes. So all of our different organs, all of our different tissues, they're all made up of cells with, different shapes, different sizes, different functions. But on a lot of those cells, there's a tiny little appendage. It's kind of like a little candle on a birthday cake, and that's called the cilium. It's actually, for more technical terms, called the primary cilium, but today let's just call it the cilium. And this cilium, literally, as you can see on this picture here, it really does stick out from a cell, and it looks like a little hair. And for a long, long, long time, first of all, people ignored it and thought it wasn't important, and literally it was only Phil's work around the turn of the millennium that we realized that actually this little hair is very, very important. And previously, we thought that this little hair, this cilium, which looks like this if you blow it up and look a little bit more detailed, is just like a radio antenna, receiving signals. So on the slide here, you can kind of see a cross-section of the cilium. It looks very technical and complicated. I don't want you to remember any of this. All I want you to remember is that what we now know is that it's no longer just working as a radio antenna. What we've now found is that this cilium is actually also able to submit and send out signals. And it does that via little packages or parcels, so to speak. And this is a game changer because, like I said, for a long time we thought the cilium was only doing one thing, receiving signals like a radio antenna. And now we're seeing that these cilia are actually also doing the opposite as well at the same time, can actually send out signals. And that's something that is very new and that we are trying to understand a little bit more of. And I just want to help you understand what this could be like. So literally, these little cilia are able to kind of bud off little packages, and then these go out of the cell, and these packages are then used for cells to communicate with each other. So cells, just like we are in the room, need to communicate with each other. They need to talk to each other so that they know if they, should be friends with each other or not be friends with each other. Sometimes they need to hold on tight to each other. And sometimes in some cells, they kind of want to stay away from each other. So all these things are very important for communication between cells. And the way some of these cells do that is by sending each other parcels. They literally are small little parcels that are packaged and have a content on the inside, a postal address on the outside. It really is very, very, very sophisticated. And so that's what we are trying to look at. So we know that these parcels exist, but we have no idea what's inside them. We know that what's inside them gets read by other cells. So it's kind of like this side of the room wants to send a message to this other side of the room, and instead of sending a text message, you're actually writing little notes and throwing them to each other. We know that these little notes are being sent backwards and forwards, but we have no idea what they're saying. And this is what we need to try and find. We need to try and find out what they're saying and what's in them. And obviously, we're talking about things on a very, very small level. So these are tiny. You can barely, you can't see them with a naked eye. In a minute, I'm going to show you some pictures on a microscope, but they are absolutely, I want to say microscopic, but smaller than microscopic, nanoscopic. So the question is, how can we catch them? How can we get them? So, and this is where you guys come in. Do you all know what your kidney does? Great. So, as you all know, your kidney makes wee. Now, your kidney also has lots and lots of cells that have cilia, which of course can explain why many people that have problems with their cilia, like in BBS, they sometimes have kidney problems. So, the great thing is that these parcels and these packages can actually end up in your urine as it's being made in the kidney. So what we can do, so here's my little kidney cell. What we can do is we can take the kidney cell, and as the urine is being made and passes through, these particles get released in it. And so all we need to do is get the urine, and then from the urine, we can collect it and harvest these little parcels. And obviously we need a lot of it. And ideally, it's best to use the first urine of the day, because overnight your bladder is making urine and it's sitting there for a while. So ideally, it's the first urine of the day. And what we then do is collect it in a little pot. We then put it in a special tube that's got a pointy end so it fits into a centrifuge. So we put it in a Falcon tube. We then spin it very, 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 very fast. And we're talking about 100,000 rotations a minute, so very, very fast. And then in the end, we will get these, we can isolate these particles. And I'm sure that by now, you've had lots of technical pictures and lots of technical info. So I thought, just to prove to you that the science that we do in the lab is not rocket science, I actually have a picture of my 4-year-old son doing these experiments and working with actually our own urine, because we've been practicing with our own urine. And what you see here are two pictures. So on your left, you have Luke filling urine into these pointy tubes. And on the right, he's then put it into this centrifuge and starting to process them. And what we get at the end is, if we then take these packages and put them under the microscope, you can see these shapes that look very much like deflated footballs. So here you have 3 pictures, and you've got the big picture, and kind of it's blown up. One of these small spots has been blown up in the corner. And hopefully what you can maybe tell is that they kind of look a bit like a deflated football. And that's actually what these vesicles look like, that these particles look like, these parcels and packages. So we know what they look like. We know that they exist. And what we're now trying to do is find out what's in them. And to do that, you have to do some very fancy molecular photography, where you take a picture of the proteins inside. And what you can see here is a picture that we've taken where we've stained with different dyes for a different component. And what we've seen so far, so this is a picture of these vesicles where you can just see one component. What I'm hoping is that you can see that different people have different amounts of this one particular protein. So these are 6 samples from 6 different people. And these are actually, this was a preliminary experiment we did. So there's 6 different people that have BBS, and they all have different amounts. So you can see in one of them there was a very light picture, and the other one there was a very strong band, and in one of them there was nothing at all. And so this was just a pilot study that we just started to have a look if there are any differences. And what we've noticed in control urine, it's much more constant, and you always get the same amount, whereas in BBS samples, you get differences. This is only one component, and we need to look at other components. And so this is where you guys come in. So what we really need to do is to look at more samples from more people, especially from people with LMBBS, to see if in the urine, in these vesicles, there is something in there that can help us explain why the kidney cells aren't working that well. And that's where you guys come in. So we're hoping that many of you are keen to help us. All we need is your we. That's all we need. We have got everything with us. And in the workshops today, you can come, if you want to come and help us, we give out pee pots. We've got gloves, we've got paper bags, so you don't have to walk around the conference with pee pots. Obviously, we've got the paperwork for your signature because we've got ethical approval to do this. We want anybody who's affected with the syndrome, any age, we really don't care. Alcohol does not affect. So that's a question I've been asked a lot. So it does not matter if you've had a couple of drinks tonight, please do. To be honest, maybe the more you drink, the more you'll pee. So the alcohol does not affect it. And again, any urinary tract infections does not affect the results either. So again, we're happy to take anything. My team and I will be handing out everything you need. We've got large pots, so they should be easy to pee in. And the key is that we want the early morning urine. So we want the first urine in the morning, and tomorrow morning we'll be in the lobby from 7 o'clock onwards, maybe a bit earlier, depending when my son gets up, and we'll be collecting them. And then at around 11 o'clock, we're going to take them to the lab in London, spin them down, because we have 3 different types of spinning down, and then we'll ship all the samples to Germany, where we'll continue the rest of the processing, and maybe by next year, if we're really lucky and Ann-Katrin and Lena work really hard, we might have some results to share with you. So with that, I also want to just, again, thank everybody in the room for helping, thank all the people so far that have donated their wee. And this picture was actually taken just a few weeks ago when we had some very special visitors to our lab. And also, just so you know, we are not doing anything on you guys that we haven't done on ourselves. So because we also often need control urine samples, we do this with our own wee as well. And with that, I just want to say thank you and ask you all for liquid gold, which is what we call your urine. So please give us your liquid gold. And if you have any questions, you can come and see us in the reception.